And with that, we will move on to our final speaker of this year,
David Sinclair,
which is David Sinclair from Harvard Medical School. He's been, of course, a pioneer in the aging field for quite a few years. David, are you there?
Yes, can you hear me?
Yes, we can hear you. Hi, David. Welcome to our conference. We're very excited to have you here. If you're able to share your slides, that would be fantastic.
All right, let me get that started.
Yes, we can hear you. Hi, David. Welcome to our conference. Very excited to have you here. If you're able to share your slides, that would be fantastic. All right. Perfect. Thank you very much. Just go ahead when you're ready.
Great. Well, thanks, Morten. Thanks, Alex, for really a one-of-a-kind conference. It's really been quite an amazing thing to see over the last 4 days. And thank you, everyone, for sticking around to the end. All right. Thanks, Morten. Thanks, Alex, for really a one-of-a-kind conference. Give me a sec. I can hear myself speak, so I'm just going to put my volume down a little bit. All right. So I'm going to start now, and here's the intro slide. All right, it's working good. So there are many hallmarks of aging, and we could think of these as causes of aging. One of those 9 causes is change to the epigenome, and this illustrates that mechanism. This is supposed to represent a nucleus where certain genes are turned on in a very specific way. The Waddington landscape is another way to think of this. Cells go from a pluripotent cell, well, a stem cell, fertilized egg, and they end up with very specific cell types. And this gene expression is very important for the function of a cell when it's young. And what the field has realized over, at least 30 years now, is that gene expression changes over time. And with new technologies, which I'll talk about, we're able to actually see these changes at, in a 4-dimensional way. And what's emerging is that perhaps one of the leading cause of problems in tissues is a loss of transcriptional patterns that are youthful, genes that are expressed when they shouldn't be in certain cells, and you end up losing cell identity. And one of the first questions, the first third of this talk, will be about trying to understand what actually is driving that process. There may be many things that happen, including things that drive senescence. And one of those, we heard Vera Gorbanova talk earlier, we think that double-stranded DNA breaks, or DSBs, are a large driver of this, and Vera's work also hints at that. To get to just a little bit more detail, if we zoom up, this is the Waddington landscape at the top to represent what happens when a cell differentiates, and this shows us 2 different types of cells, cell type A, cell type B. And this could be a neuron versus a skin cell. And there are structures called TADs, which allow the DNA to be accessible in these loops or bundled up tightly in silent heterochromatin. And we're starting to understand in a very detailed way how these loops are actually controlled and laid down, sometimes for over a century. Now the DSBs, the double-strand breaks, I'm going to show you just a few slides on evidence that this is actually a problem and actually a driver of what we can call epigenomic noise, the loss of cell identity. The other thing I want to talk about more in the second part of the talk is that there seems to be information in cells that allows for a reset. And since John Gordon did his work and won the Nobel Prize, and then the next line down, we look at cellular reprogramming, which was Shinya Yamanaka's great discovery, also Nobel Prize 2012. These studies tell us that cells have the ability to go back in age and reset their epigenome and restore different and earlier transcriptional profiles. And we know very little actually how that actually happens and where that information is stored. What I also believe is likely is that these programs that we're tapping into here in these experiments are very ancient and perhaps are involved in the regeneration of body parts, as you can see here for hydra and axolotl, and even in mammals for liver regeneration. This paper is published a long time ago, so I'm not going to show any data, but you can go check it out if you're interested. This was the start of the mammalian work to understand what we think drives changes in transcription and the epigenome. And this was the first study that in mammals really highlighted for us that DNA breaks may be a driver. We were looking at proteins moving around onto different places in the genome, such as SIRT1, and Vera and others have worked on SIRT6. But of course, there's many other factors that we think move around during aging and in response to double-strand breaks. And one of the big problems for mammals, we think, as well as yeast cells, where we first worked on this in Lenny Guarenti's lab, is that the proteins that control gene expression are also, many of them, such as SIRT1, SIRT6, are involved in DNA repair, particularly double-strand break repair. And so when we cut a chromosome, even if there's no mutation, we still can see that there are disruptions in the localization of these proteins that move around. So we wanted to test cause and effect. In this paper, we really had just a lot of correlation. So how do you test cause and effect? Well, this is the ice mice. Some of you might have heard us talk about this because we made them about a decade ago with Philip Oberdorfer, who was a postdoc at the time. And we asked the question, is the cell's response to double-strand breaks, do they induce changes in chromatin that can perhaps accelerate the aging process? And we have these mice that I'll quickly tell you about. We can also isolate cells from the mice, either embryonic or adult, and do in vitro studies. So just to jump to the chase here, Jae-Hyun Yang is the star postdoc in this study. It's in bioRxiv if you want to check it out. And this is the cell-based system where we can induce for 24 hours or as long as we want with doxycycline. We can induce the expression of an enzyme that cuts DNA, but as far as we can tell, doesn't leave mutations at the levels that we're causing them. And so we get about a 3- to 4-fold increase in the background level of double-stranded DNA breaks. These are very easy to repair cuts. They have sticky overhangs. And as it says there, we've studied these for years. We don't see any mutations of a background, no change in the cell cycle until many weeks later when senescence starts to take hold in the cell culture. This is studying a pre-senescent change in the cell. And we think that the epigenome is very important for that because we do see, as I'll show you, loss of epigenetic structures and the accumulation of epigenetic noise. And the transcriptional changes are surprisingly similar to transcriptional changes in vivo when we take, say, a fibroblast from an old mouse. So these are mouse cells that I'll show you. We've done a lot of epigenetic analysis. I'm not going to show you all this data, of course. A lot of it is already out there online. I just want to highlight some of these studies for you. There's a technology that's called HiChip that is recent, so I'll just mention it. It's like Hi-C, where you can look at the TAD structures and interactions between DNA strands, but you chromatin immunoprecipitate so you can see particular histone marks, for example, that are associated with these loops. And the kind of data that we get, which is fairly recent from Jay's work, in a collaboration with Dovetail Genomics out on the West Coast. This is really quite an amazingly high resolution map. Now, one of the loci where we see a lot of transcriptional changes is the Hox locus. These are the genes that control where your head is and your tail is during embryogenesis. And we see that this becomes deregulated quite dramatically in response to having to repair double-stranded breaks that are created elsewhere. Now, these breaks there are ostensibly, like I said, about 4 times more than the normal. But importantly, these breaks are not at the genes that we see the changes, probably because it's the proteins moving around that ends up disrupting it. And the proteins that move away, not all of them go back to where they came from. And what I want to highlight in the middle here, this HiChip data for a histone modification called H3K27 acetylation. we see that there are dramatic changes in the ice-treated cells. So to put it another way, cells that have recovered from trying to repair DNA breaks or successfully repairing DNA breaks are actually, they have a memory of that event, even if there's no genetic evidence of that. There's an epigenetic memory. And further down here, you can see that there's changes not just in the acetylation of that histone, but the methylation as well, which is these are both marks that are known to change during aging. So just skipping right along to get to the chase, the ice mice, these are the control mice if we don't express the enzyme or if we have missing one of the components of the system. They look like this. This is a mouse that's now 16 months old. These are black 6 mice. Now we turn on these non-mutagenic double-strand breaks just for 3 weeks when the mice are about 5 months old. So they're young. And they don't notice it, they don't feel it as far as we can tell. But 10 months later, the mice that experience those cuts, remember their genome is intact. We've sequenced deeply the cells and the mice, and we don't see mutations. So this is what we believe is an epigenetic change to the mice. We've done a lot of biology, physiology on these mice, and they're very similar actually to Laura's mice, the ERCC1 mice, and we do see evidence of a whole range of physiology and even down to the histological level that these mice are looking old, but that doesn't prove age, right? But fortunately, we now have the DNA methylation clock, and we can actually ask the question, are they older? And the answer is yes. What you can see there under the green arrow is that the mice that have been treated with the epigenetic disruption are about 50% older on aggregate, on average. And so that's great. That means we can quite literally say that their DNA methylation clock is older. And what I want to talk to you about in the rest of this talk is our efforts in my lab and with our wonderful collaborators to do what that green arrow is showing, is to bring the age of complex tissues back again. I think most of you will be familiar with the DNA methylation clock. There are hundreds of thousands of these DNA methylation sites in the mammalian genome, and a subset of those in the thousands, perhaps tens of thousands, depending on how you draw the line, change over time in predictable ways that you can use machine learning to actually build a clock. And that's what we did in this experiment. But the question is, can you actually get those patterns back again? Is there information in an old cell or an old tissue that you can access to reset the cell? And here we stood on the shoulders of Shinya Yamanaka's work. These are the 4 Yamanaka factors or genes. These encode transcription factors. And typically, if you want to create iPS cells from adult cells, you can use these 4 genes, you express them, and you get stem cells. And so these cells are actually going all the way back to an extremely early stage. Now, we wouldn't want to use this as a therapy. It would cause cancer throughout our bodies, and this would not be youth, this would be death. But we think now that if you use a subset of these and you use them in the right way, you can actually turn back the clock. We're also greatly inspired by this paper by Ocampo and Belmonte and colleagues at the Salk Institute that came out in 2016 in Cell. And by pulsing the 4 Yamanaka genes in this progeroid mouse model of lamin A, they were able to, you can see the mice look better, they are healthier, and they live about, by recollection, it was about 30% longer, maybe a bit longer than that. But it was a dramatic finding that these genes can actually improve health. Now, the problems with this first study include that the mice, if you keep giving them the treatment, they are predisposed to cancer, teratomas. But also, the reason it was pulsed, as you can see in the lower left, was that it was toxic. And if you expressed it for longer than a week, the mice ended up dying. So again, not a good treatment, but wonderful proof of concept. And so we were working on this actually, and we had a lot of trouble because we kept leading to oncogenesis, or at least transformation of cells in culture. But we had a breakthrough. And these 2 talented people, Wan Cheng is now a postdoc, he graduated, and Xiao Tian, who did a lot of work in Vera Gorbanova's lab, and I was lucky to have him join my lab, or our lab. They've been working on this for the last few years. 4 years, I'd say. And what they did was they left c-Myc out. So c-Myc is an oncogene. We thought that it might still work without c-Myc, and it turns out that that was the right thing to do. And so we now have OCT4, SOX2, and KLF4, or OSK for short. And Yuan Cheng built two systems where we can use tetracycline or doxycycline to turn off the genes or turn the genes on using these TTA or RTTA systems. And these are packaged within adenoviruses typically. We also have them in lentiviruses, but in the work that I'll show you, we were using AAVs or adeno-associated viruses, different types for different tissues. Now, we focused initially on the central nervous system, and partly that's because the central nervous system doesn't regenerate unless it's very, very young. So when you're young and you damage your retina or your spinal cord, it will regenerate, at least partially. But if you're an adult, your nerves are not going to grow back unless you can coax them into it. And there hasn't been a lot of success there, although people are making progress. So what we decided to do was to look at the optic nerve and see if we could reverse the age, potentially, of the eye, the optic nerve, and get it to regenerate. We did a lot of in vitro work to get to this point, but for lack of time, I'm going to jump straight to the in vivo work. We collaborated with Zhigang He's lab. The He lab is at Children's Hospital in Boston. And the model that they like to use is what's called optic nerve crush. You crush the back of the eye, and you actually can see if there's any regenerating axons using a dye called CTB. And here it's stained green, and in our studies, you'll see it's orange. And the nerve crush is actually this asterisk at the bottom where the optic nerve is dying away. But you can see in this experiment, there's some regeneration. Zhigang is actually a world leader in getting this to work. So we asked, if you put OSK and turn it on after the crush, can you re-establish some growth? And this is the result. So on the top, if you don't turn on OSK, you actually get a lot of death of the nerves. You can see a lot of the dye is now missing in the optic nerve, even heading towards the eye, which is on the left here, has died away. And we can see that in cross-section if we do that. But what you hopefully can see is that if you reprogram these eyes after the crush, we actually get a lot of preservation of the nerves to the left. And the longer we leave it, the more growth we get actually. That if you leave, so this is left for 4 weeks. If we leave the optic nerve for another, 12 weeks, in addition to that, these nerves will actually grow right back and start connecting, or at least touching the chiasm in the brain. Now, we tried this in young mice. This is the young mouse result, but we also did this in 12-month-old mice, and it also worked, which was a really piece of good news for the aging field, I think. We've gone on, prompted by wonderful reviewers, to understand what cell types are responsible. And there are a couple of ways you can do this. You can turn on OSK just in the horizontal cells or the amacrine cells, or in the RGCs, the retinal ganglial cells. And you can see the constructs at the bottom. So Yuan Cheng did this again in collaboration. And what he found was that the regeneration occurred when he overexpressed OSK in the RGCs and not in the other cells. So we think that this is a cell autonomous effect. We can actually see that those cells that received the adenovirus are those that actually survived and grew back. So we started collaborating with Bruce Cassandra's lab at Mass Eye and Ear, also in Boston. And this is the experiment. You create pressure in the eye. Probably most of you know glaucoma. It's retinal damage due to pressure. And we turned on the AAVs, and 4 weeks later asked what happened. What you can see here is on the left, the electrical signals come back when we turn on OSK. The gray is the pre-treatment and the blue is the post-treatment. And then you ask the mice, can they see moving lines on a screen? And the answer was yes. On average, the mice regained their vision about halfway back to normal. But this is quite a striking result, because in the glaucoma field, there's nothing that currently restores vision once the vision is lost. All the treatments that are out there reduce pressure and just delay the disease. So this prompted us to now ask the question, what about regular old mice? And you can see this old mouse, this one has not been treated. It's 1 year of age, and black 6 mice have poor vision by 1 year of age. We didn't go too late because you actually start to get cataract. So at 1 year of age, it was perfect. Don't mind the feces, they're irrelevant. Just the mouse is a bit worried about what's going on. But you can see this mouse is not moving its head in response to the lines. But if we now look at the mouse one of the mice that was treated with the reprogramming for 4 weeks, now you can see the mouse is moving its head. And this is called the optomotor response, and it shows that the mouse has its vision back. And if we quantify this, we can actually see that those old mice, according to this assay, have their vision back 100%, at least based on this assay. We can also look at electrical signals, and they also were restored. So this is great. It works in normal aging as well. I'll go fairly quickly for lack of time, and I want to get some questions, hopefully. Does this require changes to the clock? Now, we can ask the question, what about these enzymes that are required to remove DNA methylation? And they're either TET1, TET2, or TET3, or TDG. TDG is involved in non-replication mediated demethylation. And to cut a long story short, a lot of work, we found that both TET1, TET2, and TDG are absolutely required to restore the vision in old mice. We can also do this in vitro. We have a cell culture model if you're interested, and Shao showed that the TETs were also required for the restoration of neuronal growth in culture. So this started us thinking that perhaps the clock isn't just simply a clock, it's actually involved in the resetting process. And here we could now look at the clock. This is DNA methylation age of the cells that were damaged at the back of the eye. And you can see that the OSK treatment actually, it did reverse the methylation patterns. We can see over a time course that the methylation age goes up, and then OSK can bring it, or at least it prevents it from going too high, and then it comes back down again. What was really stunning was when we could look at, this is a heat map representing the changes both up and down of a subset of the epigenome of the DNA methylome. And this is 1,200 sites that Morgan Levine and Margarita Muir at Yale were able to use machine learning to focus on and have an aging signature. And you can see that injury changes the DNA methylome, this close-up view of the methylome, and then the OSK is able to, surprisingly maintain and reset those sites. What's surprising also is that when you look at these changes, there's a large overlap with aging itself. And so this is a p-value of 10 to the minus 200. So this is, you don't have to squint to see this correlation. So I think very few people have thought about this, but I think it's interesting to speculate why injury and aging might be similar in their effects on the DNA methylation clock. So what about gene expression? That's important for the function and may help explain why those nerves are able to regain their function in the old eye. We don't see neurogenesis, we do see just improvement in function of those neurons at the back of the eye. So if gene expression resets, what would you expect? Well, first of all, if there's no effect, the changes in gene expression would line up along the horizontal. If it makes aging worse, if it gets, even more, if it gets older or the transcription gets even less like young cells, it would look like this. And if it reversed aging, it would look like this, okay? And what we got, when we looked at the top 464 genes that were significant, they lined up in this pattern, which argues that aging has actually been reversed within this gene set. So what are these genes? Well, what's interesting is that they're involved in sensory, many of them involved in sensory perception. And also, I think about a third of them were, something like that, were involved in smell and taste receptors. So we don't know why smell and taste receptors are showing up in the eye, but no doubt many of them are involved in chemical cues sensing. But they do get dysregulated during aging and they are largely reset. And what's important about this is to realize that genes that went down a little bit with aging were reset a little bit back up to where they were And those that went really far down with aging went back to where they were. So the cell seems to know not just which genes have changed, but by how much. And then finally, some new data that we have included in the work. When you look, and when Morgan Levine's lab looked at where these methylation sites were that were changing in response to injury and aging, and asked what correlates with that, what associates, we got a number of genes, and the top three, actually you can see more than three, but the top three were PRC2 associated. So PRC2 is a polycomb repressive complex. It's important for embryogenesis and the laying down of histone methylation and also DNA methylation, and TETs are involved in that. So we speculate that perhaps part of this program, this reprogramming of the cell, involves that complex, and we're testing that now. So this is our model that OSK works through TET demethylases and perhaps the PRC complexes to change the methylation status of histones and DNA itself. And that's part of the program that actually is able to reset the cells and restore the function of complex tissue. So what I've told you is I've shown you evidence that double-strand breaks appear to accelerate aging, certainly at the DNA methylation clock level, although presumably other things are also involved. Epigenetic reprogramming can reverse the DNA methylation clock in cells and restore youthful function of complex tissues. We've done the eye, and we're now testing other tissues, such as the brain. We know that reprogramming requires active DNA methylation. And the the epigenetic clock perhaps may not just be a passive measure of age, but actually a participant in the aging process itself. And these are things that change. And I think the most important question that comes out of this that I think is just fascinating is how and where is that youthful information stored that allows for the reset? Is it a DNA modification? Is it a protein? Is it something that we're not yet considering? And thanks to the people in my lab, past and present, who've done a lot of this work. I highlighted the main people in the project, but there've been a lot of people involved who have worked very hard to get this to this point, and our collaborators along the way. Many of them are leaders in the field and have spoken at this conference. And thanks for the people who funded the project as well, and the foundations and NIH. So I'll end it there, and hopefully, if there's time, take some questions.
Fantastic talk, David. Really, really great stuff. There is about 1 million questions on Slack. So we will take maybe two or three here. And I will start with the most upvoted here. This is from Yixing He. He asks, David, what do you think are some other causes of epigenomic noise?
I will start with the...
Okay, so other causes of epigenomic noise.
Yes.
Well, I would...
David, what do you think are some other causes?
I'd speculate that treatments that cause senescence are likely involved. So this can be telomere shortening, it can be media shock, it can be very severe DNA damage. What we're testing now, and hopefully we'll have results soon, is whether reprogramming can work effectively, not just on pre-senescent cells, but also senescent cells, and perhaps reduce the SASP, the inflammation that we've heard about at this conference already.
Sounds very exciting. So the second most upvoted is actually my own question, so I'm very happy about that. Great talk, David. What do you think of an accumulation of extrachromosomal DNA as a